Hey guys. Okay, I also realized that I start every video with hey guys, so I'm gonna try to mix it up next time, but not today. So we are gonna skip through all the stuff that we've already done. You should have turned in your gel electrophoresis lab, and your PCR lab is due tonight. Please make sure you're keeping up with all that stuff because we don't want you to get behind and then have a whole bunch of things to do all at one time. So cloning is a genetically identical copy of a gene or organism. So when you think of the word clone, you probably already can kind of get to what this means. So cloning does occur naturally in nature. Bacteria does binary fission, and we talked about that in our couple units ago. And we talked about how they basically replicate their DNA, and then they split off, and we have two identical copies of the bacteria. So some plants do that from roots, and then some simple animals use budding or regeneration to do that. So mammals can be cloned through a process called nuclear transfer, and it sounds kind of confusing, but when you think of the word nuclear transfer, we're basically transferring the nucleus or the DNA to somewhere else. And so we're going to remove the nucleus of an egg cell. So we take one of those egg cells and we take the nucleus out. We're pulling out that DNA. And then we're going to take the nucleus of a somatic cell, which we know we have germ and somatic cells are just our body cells, so any other cell that's not a sex cell. And so they're going to take the nucleus of a somatic cell from the animal to be cloned, and they're going to implant that into the egg cell. And so then once that's done, the egg is going to be implanted into the uterus of the host mother, and then later on that the offspring is going to be born. So Dolly the sheep, we may have heard about her before. She's pretty popular, but she was the first famous clone. She was born in 1996, and Dolly was cloned using adult somatic cells or adult body cells. Smaller organisms like mice and frogs were successfully cloned prior to Dolly with a process that used fertilized eggs, creating twins, so that's a little bit different. But the cloning of Dolly was similar to this right here, and so we look and we see female A. And they took a body cell from female A and they pulled out that DNA. And that's the DNA we want because we're trying to clone female A. So then we take an egg cell from female B and we're going to remove the nucleus, remove that DNA from that cell. And now we're going to fuse these two together, the DNA from A and the egg cell from B, and we're going to fuse those together. The fused cell develops into an embryo, and then that's going to be placed into the uterus of a foster mother. And then when that is born, we're going to have the clone of sheep A. So pretty crazy. So Mythbuster clones have a shorter lifespan than normal. That's what a lot of people think. But the truth is that when we think about what's happening here, and we took that DNA from an adult sheep. So let's say that sheep had lived to be 10 years old, and we take that DNA and implant it and fuse it with the egg cell from another sheep, and then we implant it, and then it's born. That DNA is already 10 years old, even though it's just been born, even though this clone sheep has just been born. So if that clone sheep lives to be 30 years old, we have to add on the 10 years that the original host of the DNA had already lived. So now we really have a life expectancy of about 40 years old instead of what it looks like because that sheep's only been born for 30 years old. That's really kind of confusing, so sorry if that didn't make sense. Let me know. But the DNA had already lived some of its years, and so when it was implanted, it started at 10 rather than 0, like normally it does. So cloning of humans is not allowed in any country, which is probably good because things might get weird. But cloning of humans has been banned in 25 countries, but even in the U.S., research is not prohibited, including research in the use of nuclear transfer or other cloning techniques to produce molecules, DNA, cells other than human embryos, tissues, organs, plants, or animals other than humans. So pretty much the only thing that's off limits in this research is humans, cloning humans. Cloning has potential benefits, organs for transplant into humans, which obviously would be super helpful if we're short a kidney or there's a kidney available that doesn't match, or a liver or whatever. They could make that. Sorry, there's music in the background. So also they could save endangered species by cloning those. Cloning raises concerns, though, low success rate, so it's not often that successful, and decreased biodiversity, which we like the diversity. It's good for the ecosystem and everything, so it might not be so great in all of the ways. Genetic engineering, genetically modified organisms have a piece of another organism's DNA inserted into its DNA for the purpose of enhancing the organism. So they wouldn't make genetically modified organisms to make the organism weaker or less beneficial or whatever it is. So they're going to do this. They're going to insert DNA that's going to enhance the organism. Reasons for GMO include resistance to certain pests, diseases, environmental conditions, reduction of spoilage, resistance to chemical treatments, and improving the nutrient profile of the crop. So a whole bunch of reasons that these things are being created and the research is being done. Genetic engineering. Genetic engineering involves changing an organism's DNA to give it new traits. Genetic engineering is based on the use of recombinant DNA, and recombinant DNA contains genes from more than one organism. We're going to talk about that some more. So we've got our plasmid, and then basically what's happening, and we'll talk about this again, but the restriction enzymes are going to cut this plasmid and then allow for the foreign DNA to be placed inside the plasmid. Bacterial plasmids are often used to make recombinant DNA. So plasmids are loops of DNA in bacteria, and then restriction enzymes, which we talked about, they're going to have those places that they cut specifically, and so these are going to cut the plasmid and the foreign DNA, and then the foreign gene is going to be inserted into the plasmid. You can see that picture there, how we had our plasmid. That blue piece is going to be the inserted DNA. Genetic engineering produces organisms with new traits. So a transgenic organism, think about the word trans means across, and genic means genes, so across genes. Organism has one or more genes from another organism inserted into its genome. You can see here those sticky ends, so the restriction enzyme made sticky ends and allowed that to perfectly fit in. Transgenic bacteria can be used to produce human proteins. So gene inserted into the plasmid, plasmid inserted into the bacteria, and then the bacteria is going to express that gene, creating the protein that it would have coded for. And so a good example of this is they can create insulin, and obviously for diabetic purposes, that would be super helpful. And then transgenic plants are common in agriculture. Transgenic plants infect a plant, a bacteria infect a plant, plant expresses a foreign gene, and many crops we know now are genetically modified. Transgenic animals are used to study diseases and gene functions. So transgenic mice are used to study development and disease, and gene knockout mice used to study gene function. So basically it's saying they're using these animals to study this and honestly see what can be created and what is helpful and what is not. So yeah, that's our last slide for today. Let me know if you have any questions. Don't forget to turn in your PCR lab, your gel electrophoresis lab, and I hope to see you all soon. Bye.